Welcome to the Dairy Podcast Show. My name is Joe McFadden, and I am your host once again. Joining me today is Dr. Antonio Faciola, Associate Professor of Livestock Nutrition in the Department of Animal Sciences at the University of Florida. His overall research goal is to further our understanding of ruminant nutrition to improve the efficiency of nutrient use as a means to enhance animal production and minimize negative environmental impacts. Antonio, welcome to the Dairy Podcast Show.
Thank you, Joe. It's a pleasure to be here.
All right. On a serious note, I was just reading the news, and I have to ask, were you and your family impacted by Hurricane Ian at all?
Yeah, thanks for asking. Thankfully, no. We had very minor damage here. Luckily, Gainesville it's well inland that most hurricanes, by the time they get here, they lose a lot of their power. So thank you.
Yeah, good. I know we have a couple students that have affiliations with Florida one way or the other, and so we're certainly aware of that up here in the Northeast. And don't hesitate if you ever need anything. If whatever I could do to help, I'd be the first to help.
Thank you. Appreciate it.
My objective today is to dive into sort of rumen biology with you. And you have a nice developing career focused on rumen biology. I've read most of your, not most, you have a lot of papers, but I read several of your recent papers, and it's very compelling, some of the storylines you're developing. Over the last century, we've certainly gained a considerable appreciation of how the rumen of the cow functions. But I think as the more you study it, the more questions sort of emerge, just like anything in science. And so I have a very simple question: what drives you to study the rumen of the dairy cow? What is the interest? What is the passion? Why does that exist for you?
Yeah, thanks, Joe. I think, as you know, ruminants are such unique animals because they can convert, cellulose and fibrous materials into delicious and very nutritious foods. They're nutrient-dense foods. So the rumen plays a major role because that's where a vast majority of structural carbohydrates are going to be digested. Also, the rumen provides the microbial protein that grows in the rumen, and it's one of the main sources of amino acids to cows. The rumen is fascinating because of this anaerobic chamber that ferments and produces also gases. So there's a lot of really cool things about the rumen, and I think that's where most of my excitement lies in terms of nutrition.
Yeah, it's we just like you're heavily engaged in social media, and certainly throughout social media you get engaged with folks that have common interests. But we've recently had a chance to go out and talk to some consumers in downtown Ithaca and about their understanding of ruminant biology, and there's a lot that is missed in terms of the real sort of the strength, the value of having ruminants to produce food for us, and we need to certainly do our part in the future to really make sure that the consumer is fully aware of those benefits. One of the things that's pretty obvious when I look at your papers is that you're certainly an expert in various methodologies, and when it comes to the in vitro systems to evaluate rumen biology, for me, I can get a bit lost in terms of what's the best technique. And I just want to talk about a couple here and help you have you help me provide a little bit more understanding of the pros and cons for different techniques. And so the single flow versus dual flow continuous culture fermentation systems. Those are pretty lengthy terms, but what in the world do those mean?
Yeah, thanks for asking. I certainly developed over the years a passion for different methodologies. And even though I didn't work with those methodologies during my PhD, I did all my work was in vivo work. I saw a great opportunity to further develop those methodologies. Most of them were not developed by me. The dual flow, for example, was developed in West Virginia by Hoover in the 70s. And dual flow means that there is two flows, and one is what we consider to be a solid flow and a liquid flow, and that's very powerful because allows us to look at things like, for example, microbial population associated with liquid phase, and also microbes associated with solid phase. Whereas a single flow doesn't give us that much of details. However, it's valid methods. I think one thing that really distinguished the continuous culture from other types of in vitro that are more well known is the fact that it's a larger volume, replicates better. It's a more robust model for the rumen, and also it's a continuous fermentation. So a lot of those smaller scale in vitro systems, they ferment for 24 hours, 48 hours, etc. Here we're talking about a system that can ferment for 10 days or longer. And I think that gives a lot of power to the system.
Are there things that you can't sort of, replicate in these systems that you observe in vivo, and how do you manage that? Or
Yeah, yeah, that's a common question. Is the system it simulates the rumen, but we cannot extrapolate and say, okay, based on this diet, the cows are going to produce 45 liters of milk or something like that. So it's a very powerful tool to compare different treatments. So it can rank treatments well. It can tell you this fiber is better than this additive is better than that. However, it would be incorrect to extrapolate that to an in vivo animal because we are not actually measuring in an in vivo animal.
so, what about the rumen fluid itself? I mean, do you have certain criteria in terms of the type of rumen fluid you collect, or for how many cows? I mean, to me, this has always been sort of a mystery to me because when you read these papers where they talk about, oh, we took rumen fluid from 2 or 3 cows and we pooled that together, but there's been very little insight maybe into or description of maybe the diet or the environment or even the cow's genetics, and so talk to me about some of the criteria you use in terms of the rumen fluid.
Yeah, that's a great point, and it's something that if you really dig deep, there is very little, if any, publications actually evaluating that. So we follow the procedure we follow. We collect rumen fluid from 2 or 3 cows, depending on the experiment. We have a large herd here at the University of Florida, 550 cows, and at any given time, we have about 10 cannulated cows, depending on the needs of the department. Um, those cows, they come from the same group of animals. So we hope that they are a good representation of the herd that we have here, which is, pure hosting cows, high producing cows. And we don't know. I think that's a big question mark. We don't know what would be the best way. For example, if we were to incubate rumen fluid from single animals in each of the dual flows, we run into problems like statistical power or degrees of freedom that makes it really difficult to actually have. And we also have the variation from animal to animal. It becomes a mess. So what we try to do is try to collect a representative sample of what would be the rumen population of cows in a particular herd, and then we use that as inoculation for the system.
All right. Yeah, it makes complete sense. I mean, there's a lot of things, a lot of potential research there that we certainly could dive deep into. And one of the things I understood from reading one of your papers is that one of the disadvantages is lower protozoal populations. But what's driving that? Why is that the case?
Yeah. So we are currently evaluating that, and people like Jeff Perkins at Ohio State and collaborators also have looked at this in the past, and I think still looking at that. Um, Part of that it may be the rate of passage, that it's something that we adjust in are allowing or preventing protozoa to stay longer in the system. So they simply flow out of the rumen. And part of that, it could be that just dying inside the fermenters because it's a different environment than in the rumen. So we're trying to do some cool things to account for that, like covering, changing the colors of the fermenters. We're still working on some of those projects. Now, in the past, has some research has tried to use filters to keep the protozoa in. Uh, the problem with that is that also keeps some of the particle sizes in, or some of the nutrients don't flow out. So then, in one hand, you have better representation of protozoa population, but you have um, less ideal representation of what's flowing out, which messes up your determination of nutrient digestion, which really defeats the purpose of having a simulation of the rumen fermentation if you can't have that nutrient outflow. So it's really a fine balance. And what we are most focused on is the outputs. So we want to have adequate outflow of nutrients. We want to have adequate fermentation in the fermenters, and that's what we focus the most. So it's a lot of trial and error, to be honest with you, and that's how we fine tune those experiments.
Oh, yeah, it's very, you use a very methodical approach, which is good. I mean, I've seen your system, at least photos of it. It's quite impressive. When it comes to methane mitigation technologies, and so protozoa play a key role here in this conversation, but in terms of in vitro systems, what role do they play in sort of defining the efficacy of inhibitors of methanogenesis?
Oh, that's a very hot topic, right? It's important for efficiency, for energy utilization. So I think in vitro systems will play a big role in determining again difference in treatments, difference in feed additives. Um, so kind of like the screening process before you go to the gold standard, which is really in vivo work. So I think in vitro systems allows us to quantify total gas, to quantify different types of gases, and to correlate that or associate that with microbial population types of diets, different levels of fiber, and etc. So I think the in vitro systems really allows us to not only measure the gases, but also what's associated with that changes in gas production.
This might be a tough question, but are there examples of feed additives that were tested both in vitro and in vivo where the results don't necessarily align, or examples of feed additives where they do align closely in terms of methane inhibition?
Yeah, there's certainly some variation, and I always tell my students that every 10 or so experience we run, there is a couple that really hits the targets in terms of what we expect, and sometimes what we least expect can become in the future some of the most important findings. So there is certainly variation, and that's why we strive to do replications, to do more than one experiment. Sometimes companies approach, or even federal grants and things like. We need to do multiple experiments to validate one experiment. Often it's not enough to prove a concept.
Mm-hmm. Yeah, and I've even though I don't do any in vitro work myself, I've been advocating for the value of an in vitro data to help us better understand mode of action in terms of how these additives are potentially influencing methanogenesis. So there's a lot of value there, and look forward to seeing more come out of your group. Another question I have about protozoa is that I read a paper, you had a meta-analysis focused on the effects of ruminal protozoa on methane emissions. And you really talk about two groups of ruminal protozoa and how they uniquely influence methanogenesis. Can you explain that a bit more?
Yeah, thank you. Yeah, that's right. That's. I'm proud of this project, which is a former student in my group started working on protozoa towards the end of her PhD, and then we continue that project, and we partnered with some colleagues to look at. A large data set, and we conducted this meta-analysis, and basically when we talk about protozoa, it's a pretty diverse group of cells, and we have two main groups, entogenomorphs and isotreats, and they have very different metabolism. They also consume different types of nutrients. And isotreats, which are much larger, they are considered to be the ones that are more associated with methane production and methanogens, the organisms that produce methane. But we really didn't have a lot of evidence. And this meta-analysis showed that when we look, if we just extrapolate and say total protozoa, we're actually missing a big part of the information because entogenomorphs, even though they are very numerous in the rumen, they contribute much less to methane formation than isotreats. So looking at those populations separately give us more robust models to predict methane emissions and also to even perhaps develop target feed additives that will target specific protozoa.
Any ideas of types of additives that could influence one of these two groups of protozoa?
Yeah, not yet, not yet. We believe that some of the current additives may already be being targeted at, and we don't know. So isotreats, for example, they are known to thrive under high starch, high fermentable carbohydrates. So some of those feed additives that may be used in those type of diets may be already targeting them. So that's something that we would love to explore further.
Now, whenever I teach one a class, one of my first icebreakers I always ask, and it's very, my students probably call me a science nerd, which is fine. But I always ask them, what is your favorite organelle? Okay, and so what are hydrogenosomes, and why are they important to this conversation?
Yeah, so those are organelles that are produce they release hydrogen, and in the protozoa we believe that there is methanogens, which are those archaea population that live attached to protozoa and use those hydrogenosomes, get those hydrogens to form methane, and that's one of the really importance of those organelles.
Got it. Well, certainly a lot of research focus on methane inhibition in the near future, and certainly a lot of opportunity to better understand the role of these additives in the rumen. I want to switch gears a bit and talk about something else that's lurking in the rumen that can cause potential concern, and it's called lipopolysaccharide, or LPS. You certainly have quite a few papers sort of emerging on the study of ruminal LPS. First, what is lipopolysaccharide?
Yeah, thanks. So this is something we've been exploring the last couple of years. So lipopolysaccharides are components of the membrane of bacteria, for example, and they are present in our guts. And when bacteria dies, they release those polysaccharides, and they have uh, uh, biological function in our body, and they trigger inflammation, and that's something that people working on inflammation has been evaluated for a long time, mostly using E. coli LPS as a model for inflammation, and it's a very well defined and very well established uh, inflammation uh, uh, model for animals in general. And I was very curious about ruminants because they harbor this immense bacterial population, and they are exposed to ruminal LPS and all the time. And what does that do to the rumen or even post ruminal? So we start looking at LPS from a ruminant standpoint. How does that differ from E. coli LPS, and what kind of impacts diets will have in different types of bacteria that will produce different types of LPS? So that's in a nutshell how we got hooked on that topic.
Now, so this is something I think, I don't know, I have a hypothesis is that we've often overlooked the potential uniqueness of various types of lipopolysaccharides. And so and we've just generalized it in a way in terms of animal science of the last couple of decades. But in terms of the structure of LPS, how can that structure be unique?
Yeah, right. So depending on the bacteria, they will have different uh, lipids on those associated with those polysaccharides, and we believe that's part of the uniqueness of that compound is going to be the different types of lipids, and there is a part of the molecule called lipid A, and that may also differ between species, and it could be the reason why some LPS are considered to be more inflammatory and some LPS may be less inflammatory.
Is there any indication for types of diets or ingredients or environmental scenarios, whatever the condition, that is there? Have you observed anything where you see differences in structures?
We have very preliminary work. In fact, one of our projects was just uh, a really like a pilot study, just looking at cows eating high grain diet versus cows that are like in a pasture, and we observed some difference in the composition, but because we didn't have enough animals to really do a detailed study, I can't really have a final answer, but it does seem, and there is evidence from human literature and from other animals, that diet plays a role. And we know as ruminant nutritionists that diet will dictate at least part of the microbial population, and those microbes, they may produce different types of LPS. Yes, but not quite there yet to give a final answer on exactly how does that change.
So, in terms of LPS, there's definitely an appreciation for the fact that it can elicit an immune response. What is that? What's the biology look like? What is that? What are the mechanisms there to explain that?
Good question. I would love to team up with people who understand much more post-ruminal than I do, because it seems like the mechanism, like people like yourself, Lance Baumgart, and many others and Barry Bradford have been looking at LPS inflammation in the small intestine, even large intestine, and it seems like the mechanism it's post-ruminal. We don't know if there is any. First of all, if the receptors are there and if there is any translocation of that LPS. In other words, we don't know if that LPS really gets to the bloodstream from the rumen, or if it's just flowing out of the rumen and then becoming inflammatory agents after that. So that's what we're really trying to understand, the role that the rumen microbes play in that supply of LPS in the rumen, and also, and most important, even after the rumen.
Now, back to the structure business, is there any indication that the structure can influence the immunogenicity, the I guess the immune response? Is that influenced by LPS structure? Probably not in the cow, but maybe there's something in non-ruminants and rodents, maybe.
Yeah, well, a lot of the LPS that it's commercially available comes from E. coli LPS, and we know that that's really potent and really and causes inflammation, and people use as well-established models. When we isolate LPS from rumen bacteria, we don't get the same type of response. We've done a couple different studies isolating that LPS and using that in pure cultures, for example. And some bacteria actually like it. It seems like they use that as a carbohydrate source. So they actually thrive on that. So we want to understand which type of bacteria thrive on those conditions, and if that may be associated with other metabolic disorders like acidosis, for example. Does that create a better environment for those bacteria? We don't know. We don't know if there is cause effect there, or if it's just association. So we are just kind of starting to explore that.
Yeah, so that was my next question about the relationship between ruminal acidosis and LPS. What are the sort of the applied applications here in terms of dietary approaches to reduce ruminal LPS production? in those scenarios?
So I think it started, at least in my head, started when I started reading papers under acidosis conditions where they the authors reported a LPS concentration that was several fold higher than typical LPS levels in the rumen. So then it begs the question: is the acidosis that is causing bacterial death and release of this LPS, or is the LPS that is also available in the rumen that is causing this snowball effect, and the results that we observe, the pathogenesis that we observe in cows, is that due to the low pH in the rumen, or is that due to the LPS that is flooding the bloodstream? So that's what we are trying to better understand, if there is any association, any cause effect with metabolic disease such as acidosis and LPS concentrations.
Now, you have a paper published in the Journal of Applied and Environmental Microbiology, and I'm paraphrasing here, but you say LPS may contribute to ruminal acidosis. How does that happen?
Yeah, so in that study, we looked at pure cultures, and we dosed LPS onto these pure cultures. And as I was saying, some of the bacteria that thrive under acidosis condition, they also use LPS as a fuel. Yeah, so a substrate. So we thought that maybe it's there is something going on there, and they but we haven't been able to do many other trials, and we hope to be able to continue that work to better elucidate if not only in pure cultures but in mixed bacteria, where it's a little more realistic of what's happening in the rumen.
Now, I know you know that I've been exploring sort of total tract digestibility, but total tract permeability as well, in regards to different environmental and dietary scenarios. But we have no real way to understand what's going on in the rumen specifically. I mean, in terms of the leaky gut conversation, in the most recent podcast, we had a conversation about heat stress. Is the rumen leaky? Does it leak, and what does it leak?
Yeah, I don't know. So that's one thing. For example, in a in vitro system is difficult, right? We don't have that absorption. We have the flow out, which is already which is great. So we have an estimation of what's coming out, but we don't have absorption. So we know that things like ammonia and VFAs, they get absorbed through the rumen wall. It wouldn't surprise me to know that things like LPS and other compounds that are, let's say, metabolic active in the body also are leaking. So that's fascinating. I would love to team up with you and your students, maybe to look into more in depth on what's possibly leaking, and what is causing that, and what are the consequences of that leak.
Yeah, absolutely. I mean, lots of opportunity for sure. We have a job for the near future. There's no question about that. Now, I have this last question here is pretty open. Where do we go next to improve our understanding of rumen biology? Like, what are the what really? So you stay awake an extra 5 minutes every night because this is what you're thinking about.
Yeah, well, there's this notion that, and you read reviews, even reviews that are not very old, and the rumen still being portrayed as this black box. Oh, we don't know, there's a black box. So it really keeps me up, not all night, but maybe 5 minutes before I go to bed, thinking, what can we discover more? I mean, the great advance in biotechnology has allowed us to look at single species, and things that we often say that we know very little of the microorganisms that are present in the rumen. There is several that we simply don't know. So those are tools that, if we can translate that to nutrition, because the problem is we have a lot of excellent microbiologists. We have a lot of excellent nutritionists. Rumen Microbiology is not so many. We have several. They're great. And I think there's a lot of room for progress in that area, understanding the role that those microbes play. Now we have, of course, methane is a hot topic. Also, heat stress, as you mentioned, global warming, things like that. If we look at what a cow used to produce 10, 15, 20 years, I mean, we've made great contributions to improve that efficiency of nutrient utilization. But we can still do more. Just this meta-analysis on protozoa, for example, we could potentially reduce 35% methane by eliminating the right protozoa, for example. So there's great opportunities for sure. We really need to continue to have those highly motivated students and funding so we can actually empower them to be the next generation of rumen nutritionists, microbiologists to really take us to the next level of efficiency.
Well, thank you for that positive note. I know you've done a lot of work to support the next generation of animal scientists, and it is certainly most appreciated by everybody in our community. So thank you. I want to thank Dr. Antonio Faciola, Associate Professor of Livestock Nutrition at the University of Florida, for his contributions focused on rumen biology in the dairy cow. And thanks to those that are listening to the Dairy Podcast Show. Stay tuned for future episodes. Thank you.